When researchers sequence the RNA of individual brain cells in Alzheimer's patients, the resulting single-cell atlases reveal an anatomical contradiction. The data shows exactly which neurons die and when, but current molecular models cannot explain the order in which they fall. This graph maps interneuron survival across the progression of the disease. If we isolate the somatostatin interneuron population, a specific pattern emerges. The somatostatin population plunges downward early. These cells die off rapidly during the preclinical phase, long before a patient shows visible symptoms of dementia. Compare that to the parvalbumin interneuron population. Their survival curve remains nearly flat for decades. They survive the entirety of the preclinical phase, only experiencing a sudden drop in terminal, late-stage dementia. Biologically, this specific geometry of survival contradicts our basic understanding of cellular vulnerability. Parvalbumin cells fire at up to 200 Hz. They express calcium-permeable receptors that leave them highly exposed, and they carry the highest oxidative load of any neuron in the cortex. By conventional logic, they should die first. Instead, they survive the longest. The 8 most prominent molecular frameworks of Alzheimer's detail precisely how synapses are destroyed from the inside out. They map the kinases, the immune ligands, and the endosomal traffic jams. Yet they cannot explain why the fragile somatostatin cell dies immediately, while the highly stressed parvalbumin cell endures for decades. These frameworks identify the effectors of the disease, but lack a defined physical substrate where the pathology converges. That structure is the perineuronal net. Mapping this structure back to the atlas data resolves the paradox. Parvalbumin interneurons are enmeshed in these nets. Somatostatin interneurons, which target dendrites, lack a net entirely. Without a protective matrix, the somatostatin population is exposed to the rising neurotoxic environment immediately. They die early because they have no shield. The parvalbumin cells survive exactly as long as their net endures. The destruction of the synapse is a focal perisomatic collapse, and it is staged entirely upon this specific lattice. This 3D model shows the perineuronal net as a highly organized, multi-layered matrix, condensed around the cell body. The foundation of the lattice is a hyaluronan backbone, rigidly cross-linked by a protein called tenascin R. Built upon that backbone is the net's highest density component, a dense core of aggrecan and surrounding lectican proteins, like brevican. This composition dictates its function. First, the dense polyanion structure acts as a physical cation buffer. It manages the massive ionic fluxes required to sustain the parvalbumin cell's extreme 200 Hz firing rate. Second, the net operates as a localized antioxidant shield. The cell produces heavy metabolic exhaust, and the matrix traps and neutralizes the resulting oxidative stress before it can breach the membrane. Third, the rigid lattice physically locks the cell's receptors in place. By restricting the lateral mobility of glutamate receptors, the net maintains synaptic stability. The net is a metabolically extravagant, multi-tool armor. Its physical integrity is the only thing keeping the parvalbumin cell alive. Different research factions have spent decades isolating seemingly unrelated pathological mechanisms: iron accumulation, protease misregulation, immune complement misfires, and cytoskeletal breakdown. Returning to our structural model, we can plot these isolated frameworks geographically. They are 4 distinct biochemical vectors attacking the exact same extracellular target. The first vector is driven by lipid peroxidation. This mechanism loads the net with iron, triggering destructive Fenton reactions that cause localized oxidative fragmentation. The second vector stems from endosomal traffic jams. The neuron begins dysregulating the secretion of MMP9 and 8-AMTS proteases, which are released to actively cleave the net's aggrecan core. The third vector is an immune misfire. The neuroimmune machinery deposits C4D complement directly onto the lattice, tagging the net so that surrounding microglia will recognize it as debris and strip it away. The fourth vector is cytoskeletal. Beneath the net, destructive kinase pathways begin priming the perisomatic synapses for total structural collapse. These separate frameworks describe a coordinated siege that systematically dismantles the matrix. The destruction of the dendritic spine is heavily overdetermined, targeted simultaneously by GSK3 beta, the FXN5 cascade, and C4D immune pathways. Yet, despite the potency of these three chemistries, they cannot physically execute their attacks while the perineuronal net remains intact. The intact net functions as a physical brake on actin-myosin contraction. It forcibly holds the synapse in a rigid, disciplined configuration, repelling the internal mechanisms that would otherwise tear the spine apart. The moment the aggrecan core is cleaved and the matrix is stripped, that physical brake releases. The perisomatic synapses are abruptly exposed, allowing all three cytoskeletal attacks to engage the unprotected spine simultaneously. The diffuse molecular pathology of Alzheimer's is present long before the symptoms, but it cannot act without permission. The loss of the perineuronal net is the ultimate permission gate that allows the synapse to finally collapse. This structural failure defines phase 3 of the disease timeline, the proteolytic term. It is the exact moment the aggrecan core is stripped away. Look at what happens to the network when the shield falls. Stripped of its ionic buffer, a parvalbumin cell firing at its normal rate would quickly poison itself. Its immediate survival strategy is to downregulate its sodium channels and fall electrically quiet. The parvalbumin cell is the pacemaker for the local circuit. Silencing it removes the network's timing mechanism, causing severe disinhibition across the excitatory pyramidal cortex. Driven into a hyper-excitable state, these unchecked pyramidal neurons begin dumping massive amounts of activity-dependent amyloid and tau directly into the extracellular space. This sudden toxic dump triggers the local microglia. Sensing the rising pathology, the immune cells secrete massive amounts of MMP9, the exact protease that cleaves aggrecan, stripping the nets off adjacent healthy interneurons. Stripping just one net initiates a locked, self-sustaining loop. The loss of the net causes hyperexcitability. The hyperexcitability causes toxic release, and the toxic release drives immune cells to destroy more nets. This structural mechanism forces a complete reevaluation of the clinical Alzheimer's timeline. This timeline maps Alzheimer's from phase 1 to phase 5. The disease hinges on phase 3, when the net is stripped and the interneuron falls silent. This entire structural synthesis rests on one falsifiable prediction. In human tissue, the aggrecan core of the parvalbumin net must be definitively shown to degrade before the interneuron dies, and before the perisomatic synapses it protects are lost. If the aggrecan core is found intact in late-stage tissue, the hypothesis collapses. To arrest the disintegration of the network, the field must stop looking exclusively at the dead neuron or the upstream protein and begin defending the matrix that holds the collapse at bay.